the live stream.
Oh, right. Is it working?
Yeah.
Awesome. Okay. Cool. Welcome everybody to our theory seminar. Today we have the pleasure of hearing from.
I guess, is there like a time frame? Like, do you want me to commit to like a time frame of like, I will make sure I participate for this.
Maybe I can just say one more thing for
expected
anyone
some
other than the
intent to it,
people
but I guess I'm
speaking.
just curious,
Could you
like,
please mute?
if things get busy, like how long do people generally like?
Marna, can you mute her? I don't know how to.
Sorry,
Okay.
Susanna, could you mute yourself? Okay.
Anyways, okay. Hopefully we're all right. Right. So we have the pleasure of welcoming Aleksandra Walczak from Paris, all the way. So it's a little bit later there. So we're all coffeeed, but I don't know if your coffee is still in your system, so should be fine. Yeah, Aleksandra got her start. We used doing more like protein folding research in her master's with Jan and Toswich. And for her PhD, went to bright and sunny San Diego. It worked with Peter Wolinis on the stochastic nature of genetic switches, networks, including evolution, including dynamics of systems in the glass phase. But that was too warm, I guess, because she went to Princeton for her postdoc and worked with Bill Bialik and Curtis Gallen, among others, on information flow and gene regulatory networks, quantifying the diversity of the immune system, some really cool papers from that time. And then most recently, I guess, moved to Paris in 2010 as a permanent researcher at École Normale. I don't know how to pronounce French, I'm sorry. Co-leads a statistical biophysics group with Thierry Moreau over there. She became a director of research in 2016. And she studies the behavior of strongly coupled nonlinear systems that are out of equilibrium, mostly bio-focused. So that's why we thought she'd be an incredible presenter to hear from today. And as usual, if you have questions, feel free to raise your hand or interrupt. And with that, I'll hand it over to Aleksandra.
All right, thank you. So hopefully everybody can hear me. If I start breaking up, let me know. I'm prepared to make things better. So I'll be talking today about viral host coevolution, and it's work done with a lot of people around here. And I'll be talking a little bit about data and a little bit about theory. All the work has been done with Thierry, as Simon mentioned, but as you see, this is sort of a lot of different collaborations. So hopefully I'll be mentioning most of these people as we go along. All right, so let's start with a whodunit. Since for me, it's Friday night and the sun is setting, so this is sort of the mood I'm in. So remember our life before all of this, when we would actually go to real life scientific meetings, and a lot of them were held in remote locations. Everybody would be excited, and then there'd be a super after party. So the next morning, Joe is found dead, and next to their body, there are a few drops of blood that don't belong to Joe. So these being scientists, they cry genetic tests for everyone, which they carry out, and they find that the murderer is one of two twins, either Terry or Alex. So of course, genetic tests cannot distinguish identical twins. So they call in Detective Asia Bear, who has access to more modern technologies. And she suggests using the immune repertoire to figure out who the murderer is. So what is the immune repertoire? The immune repertoire is a bunch of cells, B cells or T cells, and these cells have receptors on the surface, and their role is to recognize foreign pathogens. So basically, these cells can perform a discriminatory task of figuring out whether the molecule presented to them is natural to your body, in which case it's fine, or it's foreign to your body, in which case you should trigger an immune response. And what happens when you trigger an immune response is that the cell, the winning cell, the cell with the receptor that recognized this pathogen, proliferates. It makes many copies of itself, which helps in making the immune response stronger. So now, thanks to sequencing technologies, we can go in, you can actually get a blood test, or in this case, as you'll see, a drop of blood, a thumb prick is enough. They sort out your T cells and they sequence these receptors. The receptors are proteins, so they're encoded in your mRNA and DNA. And due to this proliferation, when you do the sequencing, you also get counts of these of the receptors. So one other thing that I forgot to say is one cell has many receptors, but only of one type. So basically, for simplicity, the idea of receptor, the identity between a receptor and cell is a pretty okay assumption, but they do proliferate, so they make copies of themselves. So by looking at the counts, we think abundances in this immune ecosystem. So what Asia suggests is doing this kind of experiment, taking a thumb print from people and comparing the sample found next to Joe's body to samples from Alex and Terry, and then she says we can figure out we can figure out when the sample comes from the two samples come from the same person or when they come from different people. And we can figure this out just by looking at the number of shared sequences in the sample. Is there a question? Okay. Number of shared sequences in the sample. So if the two samples come from the same person, there'll be many more shared sequences than if they come from two different people, even if the two people are identical twins. And it turns out that this property of being able to basically distinguish who the sample comes from just as a function of time, just by looking at the number of shared sequences, so we can come up with slightly more complicated measures, but are basically based on the same idea, is very stable over time and actually persisted for, as we showed theoretically, for the timescale of over 40 years. So even if we studied Joe's sample 40 years later, we'd figure out that the murderer is Alex, because who else could it be in this story? And so, of course, the question is, how does it work? Why are the samples from even two identical twins so different? So for that, I need to tell you a little bit about where these receptors come from. And so they're generated. So the first thing to realize is that you have 10 to the 9th, of the order of 10 to the 9th unique receptors in your body. So receptors are proteins. If you actually wanted to encode them in your DNA directly, you wouldn't have the room. For that, it just wouldn't fit, the DNA wouldn't fit into your nucleus, because you have of the order of 10 to the 4, 10 to the 5 other genes. So this is, orders of magnitude more than the total number of genes you have. So nature has come up with a really funky way of generating these sequences and generating diversity, which combines combinatorics and randomness. So there's a certain number in your DNA, you have a certain number of templates for bits of the receptor. So the receptor is made out of 3 parts, they're called V, D, and J. And you have a certain number of V templates, D templates, and J templates, and then you randomly pick 1 V, 1 D, and 1 J. So your DNA gets edited, and you're just left with one of the templates in each T cell molecule. So that gets you, with these numbers, that gets to a diversity of an order of 1,000. We need to get to 10 to the 9th. So we have a few more orders of magnitude to go. And then special enzymes come in and randomly insert and delete nucleotides between the V and the D and the D and the J. So it's like you took 3 words, stuck them together, hope they made a sentence, but then you took the letters in between the words to sort of make it better. And you can imagine that usually when you do that, you come up with something that's complete and utter nonsense. And the same is true here. But remember, you have 2 chromosomes. So you try with the first chromosome. If that works, great, that's the end of the story. If it doesn't, you silence it and you move on to the second one. And if that doesn't work, well, the cell dies. But if it does work, you're going to get a cell that's going to express the second receptor from the second chromosome. But if you look into the DNA, or even turns out the mRNA of the cell, you're going to get this fossil record of the one that recombined but was never functional. So we'll call them out of frame reads. Oh, how does silencing work? That's a complicated question. Yeah. It basically, how does silencing work? Well, it sort of works. I mean, you do produce mRNA from this gene, so it's not complete silencing, but this mRNA is usually out of frame, so it will not make a protein. Your question is really, how does the process know that it should go and try and recombine the other receptor? And the answer to that is that this receptor attempts to be expressed, and that doesn't work. So the T cell cannot, there's a sort of selection stage that it has to pass, and it doesn't manage to pass it.
I see. So Aleksandra, it isn't that there's a protein that's made, and that protein is somehow not functional, but either in terms of conformation or actually some catalytic activity. It's actually at the translational stage where there's a failure.
No, so it can be both.
I see.
It can. So the simplest thing is it's the translation stage. So it'll just be out of frame. There'll be a stop codon, right? It can also be a conformational malfunction that it won't fold. And then there's a third possibility that it'll fold, so it needs to fold and be expressed and fold, right? So two things can go wrong on top of a stop codon, say. But even if that happens, it can be a pretty useless receptor, and that one will be functionally selected against. Okay?
That cell will be functionally selected.
Yes, thank you. Yeah, that cell will be functionally selected against. And there's a selection process, which is called thymic selection. So these cells are made in the thymus, which is somewhere here. And before they're let out into the bloodstream, they have to pass a test. That means that they're asked two questions in a way. One is, in your thymus, you're only supposed to have your own self proteins, so proteins that are natural to your body. So first of all, they ask, do they bind strongly to any, well, actually, maybe not first, forget about the order, but they ask two questions. Do you bind strongly to any of these proteins in the thymus? If you do, that's a problem, because that's called an autoimmune disease. So those receptors, even though they're molecularly fine, they get chucked away. But they also get asked a question, do you bind to anything at all? And a receptor that binds to nothing at all, although it's folded, that one also be checked away. And then we can spend the next 5 hours talking about what does anything at all mean, given that this happens in a finite time. And the answer is, honestly, nobody really understands that. But a biologist will just tell you that's the picture. Okay. But roughly, let's say that's the picture. So let's say somehow some cells make it out and some don't. And if we look at the sequencing, they actually see these out of frame reads. And for us, that's important because we can take them, we can analyze the statistics. And since we know they're out of frame, so these are like the really worst case scenario that never, they can't make a protein. We can learn something about the statistics of purely the generation process.
Because you're sequencing cells from the thymus, Aleksandra?
No, you're sequencing cells from the blood, but if you have this case where the second receptor was functional, but the first one, for simplicity, let's say, was out of frame, and for simplicity, say you sample the DNA, then it's obvious that you're going to sequence both of them. It turns out that if you sample the mRNA, you also see with a lower probability, but you also see the non-functional. Okay. So the basic idea is, because of these cells, like let's call them these cells, you actually get insight to the generation process, and you don't have to worry about selection. And there are many layers of selection that happen later. Okay.
Sorry, can I ask one more naive question?
No, this
If you're sequencing, you're sequencing the mRNA of the cells in the bloodstream, not in the thymus, but you see ones that sound like they should have been culled in the selection process in the thymus.
No, so okay,
No?
so you don't see, right? So okay, so there's three possibilities in a way. There's the possibility. So remember, you have two chromosomes. Everybody's okay with that? That we have two chromosomes, right? So if the first chromosome recombines and makes a functional receptor, this will be expressed, and the second chromosome will never recombine. So if you do sequencing, you won't actually get it because the sequencing is done in such a way, doesn't matter. Okay, maybe not interesting.
But that was DNA
second case,
sequencing, right?
Does it actually practically it doesn't matter, but it helps you think about it. Think about DNA sequencing, okay? For a mysterious reason of molecular biology, it doesn't matter whether you do mRNA or DNA. But in the simplest, let's talk about DNA. So let's do DNA sequencing, okay? So second combination is the first chromosome did not work. We recombine the second one was fine, gets expressed, and we sequence DNA, we sequence both of them. So from one cell, we're gonna get two different chunks of DNA, a screwed up one and a functional one. The third, and this cell will pass selection because of the second chromosome. So it will be seen in the bloodstream. And since it's one cell, so will this one, because it's still there, okay? And the third possibility is both of them get screwed up, okay? So that one we will not see to a first approximation. There's of course the rescue process, so you can try again, but you know, okay?
Got it. Thank you.
Now this is really cool because okay, so out we are in the bloodstream with these sequences, and now the name of the game, we're looking at these out of frame sequences, and the name of the game is figuring out what's the probability of generating a given sequence. So we want to characterize the details of this process, of this combinatoric and random process. Okay, so we take each out of frame sequence, and what we want to do is we want to say this sequence was made with this V, this D, this J, and had so many insertions and so many deletions and so on. So we can do that. But if you think about it for a second, since we've deleted and inserted random amounts and random types of nucleotides or letters at these junctions, there's many different ways we could get the same sequence. Okay, just think about the word analogy. Okay, so we can't do it deterministically, but we can do it probabilistically, and that's what we do. So we built a library of all possible scenarios that are consistent with this sequence. By scenario, I mean choice of V, D, and J, a number of insertions and deletions. We assign weights to them, and then summing over all the sequences and all the scenarios, we calculate the marginals, which are marginals of our model, for which we then use to recalculate these weights. So what is the model? The model is a model for the probability of each recombination scenario, and basically says, I pick this V, I pick this DJ, this number of deletions, this number of insertions.
Sorry, Aleksandra, I'm really sorry. If you want to just, please tell me to shut up, but otherwise I'll keep.
No, this is, I know what I'm gonna say. I actually prefer it this way.
So this is the receptor, the VDJ conglomerate that is a receptor, is that a protein complex? And somehow is it a single protein?
Yeah, it's a single protein. So you've edited the DNA, and you have a locus where you read out the single protein. Okay, again, I'm cheating here. The receptor actually looks like this.
Right.
So it has two branches. I'm telling you about this one.
Yes.
Okay, but this one works exactly the same way. we can then talk about how they co-assemble on GSAT on 2 different loci. That's what a real receptor looks like. But the story won't change. So let's just think about this. It's a single protein. It's a heterodimer.
And Aleksandra, the lengths in your nice plots, your red, the lengths are always, the total length is a constraint, it's fixed?
Roughly. It's not constrained. It's roughly similar. So again, since you're inserting and deleting stuff, this part in the middle, it's called the CDR3 region. It's important immunologically because in this picture, it's at the end here, and that's what actually interacts with the molecule, and it forms a loop. And so these can have variable length. So this picture, I'm not really showing them aligned. The deal is you can't really align them too well. Okay. There's another question. Can you sample the VD pre-VDJ? Yeah, okay, exactly. So we do know, thank you, Lauren. So we have the genomic templates, okay? So when I say what you could do is you could find the V that best matches here. The reason you can do that is because you have these templates. The problem is you can't figure out whether it's V2 or V3, because maybe V2 and V3 differ only at the end here, and that part got deleted. And that actually does happen. Okay, and especially the D gene. The D gene is sometimes it's a rather short and it gets completely eaten away. But we do have the genomic templates, and that's why we can do it. And I can tell you a funny story about what we're trying to do now with llamas, and we don't have the genomic templates for llamas, so we're trying to replace them by camels. Okay. Anybody else?
Sorry, but I just have one clarification. Do you have the templates for the individual, or do you just know
No.
VD and J? You just know it in the alleles that are present in the population? Yeah,
that's a very good question. Again, opens up a can of worms. So in databases, there's sort of the consensus gene for this allele. And for a long time, people claimed there's not a lot of diversity. And the reason for that was that because they were only sampling people, basically, lab members in North America and Europe. And so then this guy in Australia got really upset and went and sampled a lot of people in Papua New Guinea. And he said, look, at these people have all these SNPs you were completely unaware of. So there are SNPs in these genes, and our algorithm. So if we take the templates, and we see that, all of our V12s look like a V12, but in this position have a different nucleotide. We just call that as an allele. So we can call alleles computationally.
Okay.
But so there's no need to sample it for everybody, but there are SNP variabilities.
Okay, thanks.
Okay. Anybody else? All right, so here we are building the probabilistic model, right? So the probability of a given scenario is just you pick your V, you pick your D and J, and there's a reason why it's not independent, but you won't remember anyway, so I don't have to tell you. There's a number of deletions at each junction, a number of insertions at each junction, and so on and so on. So this is the most general factorizable model, consistent with the biology and self-consistent. Because what we do to learn these weights, we learn this algorithm run to self-consistently update the weights and calculate the probability of basically producing a given sequence. And when it converges, then we have the elements of the model, which are basically here. And if we sum over all these weights times the scenarios for a given sequence, times the probabilities of the scenarios for a given sequence, we get the probability of generating a given sequence. And so what you're saying here, I'm just gonna move so what you're saying here is a histogram or a density of the probability of generation for a given individual. So you basically ask, for example, for yourself, how many receptors do you have that have a probability of generation that's 10 to the minus 10, 10 to the minus 12, and so on and so on. Okay, so that's what it is. And you see that it differs by of the order of 20 orders of magnitude, meaning that you have some receptors that are relatively frequent to produce. Again, remember that, you know, you have of the order of 10 to the 9th unique ones, and you're producing of the order of really 10 to the 6th every day. So this is relatively frequent, and some that are really, really rare. Okay, so it's definitely not a heterogeneous distribution. And I could show you the elements of the model, but I won't. So that's the general idea. So the general idea, going back to our murder story, is that, okay, so what I should also tell you is that so initially we did this for a few people for whom data was available, and both the elements of the model and this distribution is incredibly reproducible, meaning that basically all of us have the same probability of generation of a given receptor. And now we've looked at the order of 1,000 people, and basically the error bars on this are very small. So this is a very reproducible process. And all that's happening here is we have a machinery that's driven by biochemistry. So it doesn't matter where you come from, what you eat, what happened to you. This machinery is basically the same. However, but it's a stochastic, so there's a machinery, which is like a random number generator, right? It's a random receptor generator. So each one of us has this random receptor generator, and although we have the same process, we're gonna generate different ensembles. So that's why each one of us has different repertoires. And that's why we can distinguish people based on that. Although we have exactly the same process, we're all different realizations of a random process. Does that make sense? You look confused.
So just to maybe see if I understand that, you're saying that the right-hand side of this plot, this density versus p-gen plot, doesn't refer to the same receptors in every human. It's just that we all have some receptors that are really highly probable. A very small amount of them. Most of them are somewhere in the middle or something.
Yeah.
Okay.
So we're all gonna, so the identity of the receptors we produce will be different, but we're gonna produce them according to this distribution. So there's many receptors that have this, well, for any of them, there's many different kinds of receptors that have this probability of generation. Like really think about each one of us as a walking random number generator.
Could we say anything about the stuff on the right-hand side of the plot? Like that it's like frequently we have like V5?
Yeah, we'll get there. I just want to, so anyway, I want to say, I sort of wanted to, this is why we can distinguish people, because each one of us will generate different receptors. So of course I'll say in a second, we'll share the ones here, but we're still going to generate some of them from here, and those will be unique to us. So about 50% of your repertoire is unique to you, and 50% of your repertoire is shared with somebody else in the world.
Aleksandra, I have another.
Can I ask a question?
Yeah.
The reason we don't, like, why is the reason we don't have the most probable sequences as the most abundant ones something like entropically? Like there's more rare ones?
That's a great question. Well, first of all, you want this process to generate diversity. You want to, this is okay, we're getting into the sort of philosophical slightly. The short answer is we don't know. But one answer would be that you could imagine, so you have to, this system protects you against pathogens. It protects you also against the pathogens that didn't exist when you were born. Right? So it has to prepare, it has to be prepared for the unknown. So, so maybe this kind of distribution is one that is good for diversity. Why are these So we're all going to produce these ones, but sorry, I think I lost the track of your question.
Like the question is like, why is not the distribution like if the most probable ones, why are they not the most abundant ones?
Okay, so first point, this has nothing to do with abundance. We'll get to that. Okay, this I should have said that. This is done on unique receptors. So for now, forget about abundance. You just look at unique receptors and you ask, what's the probability that they're generated? I mean, they're actually not the most abundant, but that's a different story. Okay.
Alexandra, there's a question from Ilya in the.
Yeah, okay. Let me do 2 slides and there's an answer to Ilya's question, which I will read. Well, maybe I won't.
But Alexandra, in the meantime, I have yet another question, which is if you can go back, just so that I can learn as much from, because you've been doing this now, you know a lot, this universal distribution. The things that would in principle alter the shape are the biochemical details of VDJ recombination.
Yeah,
is that right?
you'd need to like what could alter this distribution. So what are the elements of this distribution? What goes in? For example, the V gene usage. If you somehow produced a process that was like obsessed about using V1 all the time and wouldn't use anything else, that would alter it.
Okay.
Now, it turns out that out of the elements of this, the gene usages are the ones that vary most between people, but they're also the ones that matter the least for this distribution. Because what matters, this distribution is basically shaped by the insertions and deletions. And so let me see whether I have, I don't have this slide here. I don't have, it's like, I could probably find it, but I could show you the error bars from 9 different people. And basically the insertion distribution is really reproducible. So you could alter it again by having, say, somehow altering this enzyme. Does that, okay. So there's an enzyme that does these insertions, it's called TDT. And you could say, well, maybe I could make this enzyme such that it always adds 5 nucleotides. And that would alter this distribution. And in fact, well, in embryonic life, so before you're born, TDT is severely downregulated. So you actually produce T cells as an embryo, but without insertions. And then this distribution is different. And then when you're born, TDT gets upregulated and goes to its normal state. And in mice, we managed to trace the increase in TDT concentration based on this data. So that you really have, but to get the answer, you don't tinker with yourself, you tinker with the biochemistry to change this process. Okay, so getting to Ilya's question, which is basically about are the common ones linked to pathogens. So, okay, so we talked about generation, then there's a selection process. So there's the selection process in the thymus, which we already talked about, whether you bind to anything at all, or you don't bind to anything at all. And then these cells, or you don't bind too strongly to yourself. And then these cells go out into the bloodstream and have the job to do of finding pathogens. This is called a naive repertoire. And we've also characterized the selection process by basically comparing now, so going back to our situation, the functional receptors, or the ones from this case when this one was also functional, so the ones that actually are expressed, the statistics of that to the statistics of the generation process. I won't go into details, just trust me, we can characterize it. So at the end of the day, we can say what's the probability of finding a given sequence, receptor sequence in your bloodstream. And if we compare that to, okay, so we can then ask, if I look at myself and I look at you, how many receptors will we have in common? And I can do a theoretical calculation because how many we'll have in common is simply the probability that I generated this process and this receptor and you generated the same receptor independently. So that's pgen squared times the square of the selection factors because we also had to select them independently. So that's the model prediction. And the dots are the data. This is the number of shared sequences as a function of the number of people. So this is how many receptors are shared by 2 people, 10 people, 100 people, and so on. So you can see that at the level of unique receptors, again, we can predict exactly how many receptors you'll share with 1,000 other people in the world. Okay? And again, the ones you share are the ones that have a high probability to be generated. So they're the ones from over here. And these are the ones that are specific to you. Okay, so there's a question. Isn't there some recent evidence from B cells that there are different individual generation rules in B cells? Is this a difference between T cells? Okay, I don't know. I would have to read it and tell you about that, so I can get back to you about that. In principle, B cell generation works exactly the same as T cell generation. There's a bit of yeah, and we've looked at it and we can characterize it too. So from any data I've looked at, I don't see any difference. So I'm curious to see what this paper says exactly. Okay. But I don't know it. So, okay. So you may be worried at this point that, so we're generating all these random receptors. You may be worried that as humanity, maybe we've generated all possible receptors that this machinery basically can generate, right? So have we exhausted diversity? And the answer is no. This is what we've sampled. Of course, when you do these experiments, you don't drain somebody of all the blood. They would die. So you sample some fraction. This is the difference between what you sample and the whole thing. We could extrapolate with the model. We can also extrapolate what is, given the size of the actual repertoire. So this is as a function of people, how many, so basically adding one person, how many new receptors do you add to your pile? And so if we were saturating, this would saturate, right? If we were exhausting diversity. So you can see with the number of people, 10 billion today that we have on the planet, we're nowhere near saturating this. So this process really has a huge amount of potential to generate diversity, and it generates it. Yeah, what did I want to say else? Yeah, at this point, somebody will ask, well, you can take a corpse and you can drain a corpse. So nobody's done that. There's probably good ethical reasons why you don't drain corpses. But I'm just saying it right now because it's a question I often get. Oh, why couldn't you just study dead people?
What's the concern? Why wouldn't like a pint of blood be enough? Be a good sample.
No, because, okay, so it's enough to get the frequent ones, and it's enough to get some sampling of the rare ones, right? So we're fine. So we can learn the statistics of the process, and we can extrapolate from the model to all of this, right? But, I'm just saying that people often ask me why, to sort of, for various questions about model validation and so on, why couldn't you actually sample this from corpses? And I'm just preempting the question. Okay.
Aleksandra, just to ask one, just to understand in some senses your intuition about this, to see more unique sequences, is it wiser to sequence more blood from the average North American or European person, or do you want to go and try to sequence the same one pint from someone? Yeah, what is actually?
No, at this level, it doesn't matter.
Mm-hmm. I see. So
I mean, yeah, John Doe is not going to be more similar to Jane Doe than he is to, Pankaj, whatever, right? Or whoever you want to name. It really doesn't matter, is the bottom line here. We're all the same. This is like, you know, a UNESCO message. Okay.
Seems like a funny way for an immune system to work, because it seems like it would place evolutionary pressure on a pathogen to deal with only those very rare sequences, right?
So Ilya's question and Seb's question. Okay, so our intuition, I think for many of us, is that these sequences that have a high probability to be generated should be there against some pathogen, right? Something that's been along with us for the ride and co-evolving since early days. And so, of course, we don't know what our favorite pathogen is, but we do have a list of various pathogens here, and we know for some of them the T cells that recognize them. So we can evaluate the probability of generation of these T cells that recognize these pathogens, marked in different colors, and plot it. And in the background in gray, you see the original pigeon distribution I showed you before. So you can see that against, with the huge caveat that we can only do this for the pathogens that are in databases, and arguably there's no like basic worm in there, they all fall on the same distribution. So T cells that recognize pathogens are in no way different than your average T cell. Now, as I said, with the huge caveat that maybe we're missing, you know, the set of basic pathogens we've co-evolved with. But the other argument could be we haven't been co-evolving for that long with them. So that's the data, okay?
Sorry, Aleksandra, I'm going to have to ask you to repeat this. For example, purple, influenza A. This is saying if I chopped up the genome randomly of influenza, how do I make the influenza A distribution? Sorry,
okay,
I'm stupid.
So you take, so people know various T cells that recognize various epitopes from influenza A. You make a list of these T cells that are known to recognize various epitopes from influenza A, and you evaluate the probability of generation. And you ask whether the distribution of the probability of generation is somehow skewed to the more likely ones or the less likely ones. And it's not skewed in any way. Okay, that's the statement. Again, with the caveat that, you know, maybe there is something that would look like this and be somewhere over here. It's just not in the databases. Okay. So I think that answers Ilya's question too, right?
Just a naive biology question, which is, do these receptors have any function besides recognizing pathogens? Is there any known function besides recognizing pathogens?
This is a huge function. This is like, I mean, okay, I mean, first approximation, no, they're busy with this primary function of this. Then the receptors themselves don't take part in cell-cell communication, but there's different receptors on these cells. So there's later like there's quorum sensing involved. These cells, you know, communicate and stuff like that. But yeah, I mean, their function is to recognize pathogens, and they're busy with that a lot.
Yeah, I mean, I guess I was wondering these like very high probability sequences, you know, if they had something else besides pathogens. But,
Well, okay, pathogens is a simplification of antigen, and antigen is any molecule.
Yep.
I mean, their purpose is discriminatory, to discriminate between stuff that's natural to your body from stuff that's not natural. Or so, for example, when you get, you know, when you have tumor growth, in principle, they're able to detect it. So this whole thing about, you know, we have many tumors all the time, and most of them actually are detected by our immune system. It's also this. So, I mean, pathogen don't think just viruses, think everything that can go wrong.
Great.
Alex, should I think about the the number VDJ, V times D times J, as the number of pathogens that the human body is co-evolved with, or no?
It happens to be I mean, no, I mean, no, I wouldn't. We don't know what's the number of pathogens. I mean, okay, we don't know anything about pathogens. This is sort of, when you think about this system as a sensory system, pathogens are, you know, the environment, and we don't know anything about the statistics of the environment. So if anybody, I'm not going to talk about this today. I wasn't even planning to talk about this today. If anybody's interested, I'm supposed to be giving a colloquium at U of Chicago in March, and I'll talk about that more then. But the bottom line is, part of the problem, as in any sensory problem, is that we have very little idea about the statistics of the environment. Like, we can, you know, I guess for the visual system, we can take images, natural images, quantify some statistics, but already for olfaction, what are the statistics of possible smells, right? So here, what are the statistics of pathogens? What are the pathogens out even now? No idea. And if you ask this question on evolutionary timescales, I mean, it gets even worse. And remember, I think that the key caveat is, and I'll give an example of this system in principle does protect us. I mean, in some, let's say, magical rather way, against things that will not exist when you were born. Like COVID. We'll get there. Okay? So
Aleksandra, is there a version of the question that you could make another colored distribution for self? Like
yeah, but you'd have to get statistics of self.
Yeah, right.
Okay, and remember this distribution. Okay, so there's other variants of this question you can ask. One is the distribution for naive cells. So okay, so you get naive cells, the ones that are in the periphery, in the bloodstream. Then what happens is, after an infection, some fraction of cells that responded to infection are set aside and put into a special homeostatic compartment called memory cells. So you can ask whether memory cells have different statistics than naive cells. The answer is no. They follow exactly the same distribution, which in second thoughts makes sense because memory cells are just sampled from naive cells. But you could imagine that they're sampled in some special way, but they're not. They follow the PGen distribution follows exactly the same distribution. Yeah, we've looked, and actually Francesca, I think, is here, who's been looking at this. We've looked at different developmental stages in the mice of when these cells are developed. So these cells have their own developmental pathway in the sense of they go from a pre-T cell to a mature T cell. And in the mice with collaborators at the Weizmann, we've sampled different stages of this and saw how the distribution changes. And it doesn't change that much. The selection pressures change a little. So, yeah. But at the end of the day, remember this, yeah, these are unique cells. And the cell, we're asking what's the probability of generation of these cells. We're not even asking what's the probability of seeing them in the bloodstream. So those two things, and so once we stop talking about unique cells, that's where whatever happened to you matters. But there's a lot of other sort of problems that appear. I'll talk about some of them in a second. Okay, but so for now, we're just ending with the notion that we're all random number generators, okay? And okay, so we did this sequencing and we discovered that Terry is maybe not the murderer, but they have COVID, as does Pat and other conference participants. Okay, so now you're way more knowledgeable than the slide says, but the conference participants can all say, oh, but you're looking at T cells, and we all know that COVID test antibodies, and antibodies are B cells, because everybody cares about B cells in terms of COVID. What do T cells have to do with it? So now I think it's more, so when we first did this study, that was a big thing that people said, oh, T cells have nothing to do with COVID, it's just B cells. So this is one of the first studies that showed the contrary. So we are going to talk about T cells. Okay. So Terry and Pat, before going to this conference, went to another conference, which was a skiing conference. And they, of course, partied as they always did. And then they came back with COVID. So they self-isolated. This is early days. This is February 2020. And then donated blood at days 15 and so on until 85 post onset of symptoms. And so we looked at the T cells, and now we're going to look at abundances. And so we're looking at abundances of different T cell clonotypes as a function of time. This is what the raw data looks like, okay? It looks like a mess. What you want the data to look like is you want a peak. So the first thing we did is we did PCA on this time-dependent data. We just asked, can we find some clusters? And so we identified these 3 clusters. The purple one is the nothing happens cluster. That's a big part of cells that just don't care about this infection. And then there are 2 clusters, the green one, which is the one we expected, with a peak at day 15 after the onset of symptoms, or earlier, since we don't have data earlier. That's what you expect, right? By day 15, they were fine. And then another mysterious peak at day 37, which we don't understand. Other people have seen since, but we still don't know where it comes from. And so what you should also know about Terry and Pat is that both of them had mild symptoms. Nothing bad happened to them, okay? They were not hospitalized, no ICU, so it's written here, right? Your sort of your average thing. All right. However, so we didn't really trust this PCA analysis because we know there's a lot of noise in this system. So if you take data from,
can I bother you?
Yeah.
I didn't quite get what were those plots showing? What's average clone trajectory mean?
Oh, this is just as a function of time. So you take this, each one point here is basically receptor in the previous language we were looking at, and you see how the frequency of this receptor, normalized frequency, changes as a function of time. And you average, so you average all the yellow ones, and it's an average over all the yellow clones.
And Aleksandra, I just want to understand a little bit more about the actual, so abundance of immune cell types of each type, meaning you're getting lymphocytes, myeloid, you're getting and this is just abundances of all those different cell types, or this is abundances
No. So we're still in TCR, right? T cell receptors. So think about what this is you take a given receptor, really a given receptor sequence, but okay, there's a lot of technicalities. Basically, when you do the experiment, you'll see now that each T cell receptor can be there in 1,000 copies, 20 copies, and so on. And that's what abundance means. Okay, so it's a proxy because there's various normalizations, things that I really don't want to get into. It's a proxy for, the number of cells in the sample. So that's why I sort of use receptor cell interchangeably. Okay, so if we look at data from Terry from the same day, so we have this ×2 means we have replicates everywhere. We basically have 2 samples from the same person taken at the same time. And so if we look at the number of a given receptor in the first sample versus the number of the same receptor in the other sample, that's what's plotted here. So this should be a straight line, okay? This is the same, these are replicates from the same person at the same time. But you see there's a lot of noise. So there's some receptors that in one sample are there in a single, you see them once, and the other sample you see 100 times.
So biological replicates or technical replicates? You're taking the same sample and putting it into two lanes, or they're two samples?
No, no, actually, I think the technical replicates here, that you draw blood once, and then before you do anything else, you separate it into two parts. But you basically you prick once. Again, for ethical reasons, because there's limits on how many times you can torture a person. Okay. There's another interesting question. What would happen if you took blood here and here at the same time? Again, nobody's done that. And I would love to see that experiment. But let's assume it's a well-mixed system, which is what everybody seems to believe. Okay. But see, a lot of noise. So where does the noise come from? Well, of course, it comes from sampling. But it also comes from biological noise. We're looking at mRNA here. We know mRNAs are produced in bursts. So we could actually, the number of reads we see in our sample is related to the original frequency of these cells in the sample. And I'm not going to show you this, but from other experiments, we know that the frequency of different receptors or cells is a power law distribution with long tails. But we're getting from this underlying power law distribution to the distribution of reads in the two samples. And we have to deal with both sampling noise and long-tailed noise coming from biological processes. So we can put these two noises together and basically characterize them using the same name replicates, characterize their properties, and learn a noise model. And once we have a noise model, we can go in and use that to actually figure out what a response is. Okay? So that's the important point there is that we actually do need to calibrate the noise because it is very noisy. So then we say that the frequency at the later day, say, compared to the frequency the previous day is related by some expansion factor. Not all cells expand. As we saw, we had the purple one. So there's some fraction of non-expanding cells and some fraction of expanding ones. And then we use the posterior to identify significantly expanded clones. So after doing that, we get back what we had roughly before from this rough PCA analysis, but now with a lot more confidence. So we get the peak at day 15, and we get the secondary peak at day 37. And also what we do have is we have data points for both Pat and Terry a year and 2 years before COVID was identified on this planet. So we have the sampled repertoires. So we can also look and identify responding cells that, according to this statement, are responding earlier in time, and we see that this is really a peak, that there is a large expansion. And we also find by looking at the ID, so then we get lists of T cells that responded, and we don't find, and remember we have these databases with T cells that are specific to a virus. We can't find any T cells that are specific to known viruses. So this sort of is good news. So they're probably new. Now, going again into, so now then we restrict ourselves. We also had a separation here done into the naive and memory cells. So I already told you that naive are the ones after the infection. And so we see that after infection, they have stable post-infection memory. However, they also, and this is the interesting thing, so this is okay, this is interesting that you actually have stable memory from T cells, but the more interesting thing is that in the years before COVID, there are cells that respond. So these people had pre-existing immunity. They had T cells before, a year and 2 years before it struck, that are known, that we say are responding to SARS-CoV-2, okay? So they had potentially pre-existing immunity. And I get to that in a little bit more detail. This is maybe not so interesting about how it changes. Now, what you could be asked about is how these cells are COVID specific, right? All I'm sort of telling you so far is that these cells respond. They sort of expand when COVID hits, but maybe something else hit these people. Maybe they expanded by chance. We actually know they expand by chance also. So it so happened that Pat took part in another study 20 days after the onset of symptoms, which was like a fishing experiment study. Okay, so what's the question? This is maybe my ignorance, but I was surprised that the clone expansion, yeah, well, it is what it is. It's actually a pretty strong expansion. So there's two elements here. First of all, a small fraction of your repertoire responds to anything, even if you have a chronic infection, even if you have HIV, the fraction of your repertoire that's controlling HIV is really, really small. And the second part is, yeah, expansion is relatively limited. There's an exception to this story, which is CMV in old people. So CMV is a virus that I have, and it is linked to life expectancy, but this virus doesn't do anything. It just sits there. However, the immune system gets obsessed about it, and it controls it. It basically, although the virus wouldn't do anything, it spends a lot of resources making sure it's under control and doesn't grow. And as you get older, more and more cells are dedicated to this task. And if you get to the age of about 80 and you have CMV, basically the majority of your repertoire does nothing else but controls for CMV, not growing. And then you die from the fact that you don't have any T cells or B cells left to do anything else. Okay, anecdote for how the immune system works. So Pat's taking part in a fishing experiment study. So what they do is they stick a COVID epitope on a molecular fishing rod, and they stick it in Pat's blood, and then they pull out the T cells that bind to it. Okay, this is called the tetramer study. And so the deal is that you can only stick one SARS-CoV-2 protein from one epitope. You stick one epitope on it. So it's not like you're not going to figure out what all the T cells that respond to SARS-CoV-2 are, but you're going to figure out specifically to this one protein what responds. And so they pull out the T cells, and then we take these T cells and we match them against the ones we identified as responding. And so these are the green dots, basically. These are, well, all of them are the ones that, we first matched them against the ones that we sampled in Pat's repertoires at different days. So that's all the dots. And the green dots are the ones that we independently called as responding. So you're saying that first of all, we're reproducing the contracting trend as a function of time. And we're basically getting most of the high frequency clonotypes. So we have a patient-specific way of figuring out COVID-specific clonotypes. But mostly it just gives us confidence that what we're capturing is actually COVID-specific, okay? Because this is a direct test. Okay, so then Harlan Robbins.
Alexandra, that was the first peak, not the second peak.
Yeah. Okay, so first of all, this is only for one epitope, right? This is for one COVID epitope. What we're capturing in our experiment, so we're capturing many more responding clonotypes than marked here, right? But yeah, all of them to this specific one. The second peak is really mysterious, honestly. So this idea is that maybe this is a secondary B cell response that calls for a T cell response. That's sort of a probable explanation. But nobody's really pinpointed that. Yeah. So
sorry,
here
I have
we're
one more
also,
question.
yeah.
So with this experiment, to what extent do you think, like, are you identifying all of the, or like essential, like most of the clonotypes that are responding to that particular protein, or like how good is it at sampling all the fraction of the clonotypes?
You mean the tetramer experiment, the fishing
Yeah.
rod experiment, what it pulls? I mean, it probably doesn't get every. I think it's pretty good in the sum. Okay, given that you're putting this, you're not putting this into a person. You're putting this into a test tube, right? So you've already sampled blood from somebody. Given that, you're probably pretty good at pulling out whatever will bind to it. The bigger problem is that it's really one single protein.
Yeah.
Okay. And this is actually an interesting point in general, that, in your body, when it presents cells, molecules, proteins from, say, SARS-CoV-2 or anything else, it presents many proteins at the same time to your immune system. And your immune system actually goes after all of them. And that's what we see here. We see a very diverse response. I mean, not in this experiment, in sort of our calling of proteins. And so these experiments cannot handle that.
Aleksandra,
So
I just want to ask you that your CMV, maybe I'm even getting the name wrong, that anecdote, it seemed to give a sense that there was some overall limited resource.
Yes.
What is this? What is that?
So, okay. So it's
is it like size of your thymus or something like this?
No. Okay, so there's an element of the size of your thymus too. Let me start with the sort of mechanistic, let me start with the phenotypic explanation, and then you'll probably ask why that is, and I'll probably say I don't know. So the fact is that independently, the naive pool and the memory pool have homeostasis. They have control of the size of these pools. Now, this is not like, how is that controlled exactly? I don't think we understand. We do know that the relative size of the memory pool grows with age, and the size of the naive pool contracts. What we do know is that your thymus does contract with age, so your thymic output goes down. People used to think that by the time you're past puberty, game over, you're not producing any more T cells. That's not true. You produce new T cells until you die, and no matter what age you die at, but you do produce less as you grow older. And so that definitely influences it. And your memory pool grows older. I mean, as you grow older, you definitely, to allow new cells into the memory pool, you have to kill some. But then I guess, there is some room for expansion later on in life. Exactly how, okay, so again, mechanistically, people know that homeostasis is established through cell signaling and cytokines. But what sets it, it's in models, it's usually some sort of self-consistent about, just imposing that the integral is that. In practice, I don't think people really understand what sets it. So, I mean, beyond saying it's cell-cell communication. And whether there's some internal resources that control it. So there is fighting for cytokines. So I think that is probably a big thing. That you need, so also, okay, you also need you need non-specific signals, which people sometimes call tickling, to keep these cells alive. So they don't proliferate, but they'll just divide. So the concept of a difference between a clone and a cell is also important in all of this, because it's not exactly the same cell that you've had since it was first produced, but it'll be the same clone. So there's all these processes that basically contribute to that. So it's complicated. Okay, anybody else? Okay, so now so what Harlan Robbins did is he did a large-scale fishing experiment where he basically took a lot of SARS-CoV-2 epitopes on fishing rods and put it into the blood of basically put it into many blood samples. But it's the same idea. You pull out then the different T cells that respond to the different epitopes, and you know what epitope it responds to. So these are the green dots. This is called the mirror experiment. And then we took our blue dots, which are the ones we identified in PAT, well, in this case in PAT, and mixed them with what they identified in their experiment and built similarity networks. So we basically connected the T cells that differed by most one amino acid, and we built clusters. And this way we were able to identify both similarity clusters, as you see here, but we were also able to link, for some of these clusters at least, that had cells coming from here, what SARS-CoV-2 epitope they correspond to. So now we have a bunch of T cells that are similar, not necessarily the same, and we have a candidate epitope to which they respond. Okay, not really a candidate, because from this experiment we know that they responded. So I'm not going to talk about all clusters. I'll talk about this one, which is Pat's favorite cluster, because it makes up 21% of Pat's response to COVID, to the COVID infection they had in general. So it's a big thing for them. And this is the COVID epitope that it responds to. And it turns out that T cells from this cluster that are specific to this epitope were already there in Pat's memory a year before the infection. And if we make a one amino acid mutation in the epitope, in the SARS-CoV protein, we come up with a common cold coronavirus. So our suspicion here is that Pat got the cold a year or two before COVID, and maybe that's why they had a relatively mild infection when they got SARS-CoV-2. So this is a hypothesis that we're trying to verify now with people who ended up in ICU and see. Of course, we don't have the data from a year before, but we have some idea of T cells that are specific against common colds and so on. So
Sorry,
we're not there yet. Yeah.
Yeah, I might have missed this. Are you sure? Are you sure that these are like memory T cells? Like how are you distinguishing?
Yeah, so when you do the experiment, okay, so you have a cell, it has the receptor, it also has other receptors. One of these receptors it has is some of these receptors are basically like flags. So when a cell becomes a memory cell, it expresses a specific type of receptor. So when you get these cells, you can fact sort them based on these receptors, and that's how you know the memory. And this is an art, okay? But we trust that here the experiment was really done well, and we trust the memory.
Cool, thanks.
Okay, so that's my data story. More or less, I can tell you one more thing about data. So if we go back to these plots about sequence sharing, so this is what I showed you before, the number of shared sequences as a function of the number of people and T cells. We can do the same thing for B cells. So Maria did this is a recent paper. And we can do the same for B cells in healthy people, and we can also predict. So this is back to Lauren's question about whether B cell generation is different. So to the best of my knowledge, it's not. And we're very good at also predicting the number of shared sequences. But if we look at B cells in people who recently had COVID, they share many more B cell sequences than predicted by chance. And we know that COVID converges the repertoire in a statistically visible way. So we know that people who get COVID, they have very similar B cell responses. Not so T cells, and I can explain why, but for B cells, they have very similar responses. And we're able to see this in this kind of analysis. Okay, so this is really the end of my data story, and I don't know what to do. So I've based roughly, I have 15 more minutes on what you told me. So I can happily just answer more questions, or I can tell you, and I don't really know if I have time, about the theoretical idea.
Something I would also say is that, I mean, so now we have 22 people here, which is still a good number, and certainly people who will watch replay. Karna, Simon, would you rather move on to the sort of soft philosophical questions or that we like to ask our speakers, or should we let Aleksandra continue for sort of 10-15 minutes?
I mean, we can do that. I mean, if there is a section of, I'm happy to stop.
I don't know. I'm fine with if you have like another short story to share.
The thing is, I'm worried it's not short enough for 10 minutes.
Okay. Then let's stop, and we can always, if Aleksandra is up for it, always invite her again to teach us.
Yeah, I can do this again, and now I know how. I know
we asked a lot of questions, so I mean, but that's a good thing.
No, it's cool.
That's a good thing. Okay, good.
Format. Cool. Yeah. So that was really fun.